Well, hello there. Who's ready to do part 2 of the State Diffusion Investigating a Cell Lab? Here are your materials for today. You will have your cup that you worked on in the last lab when you made the cell. You're going to have a test tube rack, and we're actually going to use 2 test tubes today. We will need this solution that we used yesterday for the inside of the cell to verify its contents. We're going to have a dropper pipette like this, and we'll also have a test tube holder that we used the other day. So what we're going to do first is we're going to examine our cell contents, and we should notice some color changes in there. You are going to label your diagram that is right here for final state, and then you're also going to mark on the back on the first data table any initial color for inside and outside the cell, and then the final color inside and outside the cell. Then you're going to move on to looking at the contents outside the cell, so in this liquid here. So you're going to use the pipette to gather a small sample. So if you squeeze, dip it in, and then release, you should see some liquid in there. Okay, you can see the line there. Then you're going to place that in one of the 2 test tubes. Okay. After you do that, you're going to add Benedict's, and then you're going to use the test tube holder to place that into your hot water bath. You are also going to do the same exact test with the solution here, okay? And remember, this is what we put inside the cell, and the color changes there we will notice for the inside the cell, and the outside the cell will be the color changes from the liquid that you take here after you do the Benedict's test. When you're done, you're going to rinse out both of those 2 test tubes, and then you'll put your cup in the place that your class had them saved.